During the last video, we talked about how Mendel was able to use Punnett squares to explain the patterns he was seeing in his garden for traits that seem to have two different versions or two different alleles. He was able to use these Punnett squares to come up with some laws or theories of segregation. Mendel's theory of segregation states that parents may be homozygous for a trait or they may be heterozygous for a trait, and that they do have two alleles or two versions of the code for the different traits, one from mom, one from their dad. When gametes are formed, remember we talked about meiosis last time. So during the formation of gametes, we're going to go from diploid to haploid. And when we go from diploid to haploid, those alleles for those traits segregate or separate from each other. So half will go to one gamete, which is an egg or a sperm, and the other half will go to the other gamete or egg or sperm, which means that each one will get a different copy of the allele. And sometimes that will lead to completely different alleles in each gamete, and sometimes it's still the same allele. So if it's heterozygous parent, they're going to get one copy of each allele in different gametes. If it's a homozygous parent, you're going to have the same allele anyway, right? But either way, one copy of each allele goes into a different gamete. So they segregate or separate from each other. That's Mendel's theory of segregation. You guys do need to know that one. Okay, so then he thought, well, what about more than one trait? If we're looking at more than one trait, are these traits tied together or are they separate? So we decided to run some Punnett squares that are a bit more complicated. They're called dihybrid crosses. Di meaning two. He's going to look at hybrid of two different traits. So in this one, we're going to look at tall pea plants, tall versus dwarf pea plants, as well as purple versus white flowers. And you can tell from the lettering here that tall is dominant trait and purple is a dominant trait. Okay, so we're going to look at dihybrid. Hybrid means heterozygous. So we're going to look at heterozygous parents for each trait. So one parent, I'm going to put their alleles up here. They're possible alleles for each gamete combination. Okay, so after meiosis, they have the possibility of producing these four different types of gamete combinations for these two traits. Both could be dominant. One could be dominant while the other's recessive, or they both could be recessive. And the same with the other parents. So we're going to cross this parent and this parent. Okay, let's look at what happens. We're going to have this gamete and this gamete come together for fertilization right here. So we're going to go from haploid gamete and a haploid gamete to produce a diploid offspring right here. Okay, and what we end up with would be a dominant, homozygous dominant for tall, and homozygous dominant for purple in this individual. And we're going to do this each way across. So right here, we're going to have big T, and big P, little p. So homozygous dominant for tall and heterozygous for purple. Make sure you say the different genotype vocabulary terms as you go across here. It'll help you get used to it because you will need to know that as well. So right here, we have a dominant allele for tall and a recessive allele for tall. And then you have dominant for purple, dominant for purple. So we have heterozygous for tall and homozygous dominant for purple. And you can do that all the way across. Remember, these are located, you can get my PowerPoints in the content section. So you can pull this up and go through it. And every time, if you blow it up to a big screen by hitting, let me show you this real quick. This is kind of important. You can go right here, you guys see this? And you can hit this button on the PowerPoint and it turns it into interactive mode right here, where then you can just hit the arrow on your keyboard and go through the different steps. So you can try to guess it yourself or try it on paper as well. But make sure you practice these dihybrid crosses. So here's what we end up with as we go across. I have this one colored in white because this is going to be a tall white flowered pea plant. tall white flowered. This one here in the orange letters, those are actually short, and there's only one short white flower. So anyway, Mendel looked at this dihybrid cross to look for patterns. And here's what he saw every single time. We're going to look at the phenotype results. Remember, phenotype means physical. So what does the plant physically look like? And he ended up with a ratio of 9 to 3 to 3 to 1. 9 tall purple plants. So that's those are both dominant traits. 3 tall white plants. We have 1 dominant trait and 1 recessive trait. 3 short purple plants. We have 1 recessive, 1 dominant trait, and only 1 short white plant, which is going to be the 2 recessive traits. And he did these dihybrid crosses with a whole bunch of different combinations of the different characteristics we talked about in the previous video for these pea plants. And every time he came up with the same results. So that led to his next theory, which is Mendel's theory of independent assortment. And that's going to look at how different traits will separate or segregate into different gametes. What he found out was that every allele segregates independently of other alleles. This means that one gamete has an equal chance of having any combination of alleles. So let's look down here. That's kind of confusing when you read it for the first time. So I have them colored for different parents, the parents of this individual. Okay, so one parent gave this individual down here 2 dominant alleles for these traits A and B. And the other parent gave this individual 2 recessive alleles for these traits A and B. Now, just because the dominant alleles came from one parent and the recessive alleles came from the other, doesn't mean they're more likely to be together. So yes, these 2 will come off into some gametes. Some gametes will have only the alleles from one parent, and some gametes will have only the alleles from the other parent. But that's just as likely as having the alleles mixed up. So any one of these are all different possible gametes that could be formed from this individual. Any one of these is just as likely as another. So there's no favoritism here with which versions or which alleles might go into a gamete. They're all just as likely. So you can see this for both egg and sperm. Okay, and that's Mendel's theory of independent assortment. So we're looking at more than one trait. Okay, let's look at our genetic terminology. We have already gone through a whole bunch of this, even from the last chapter. So one thing, these are homologous chromosomes right here, and these are homologous chromosomes right here. Homologous meaning they're the same, they have the same gene loci. They code for the same genes. They're about the same size. That's what makes them homologous. Now, they may have different alleles, however. So an allele can be either dominant or recessive. For example, even though these are homologous chromosomes, they do code for the same genes, but that might be a different code for those genes. So this chromosome right here has a dominant allele, and then the other chromosome has a recessive allele. So they're not exactly the same. Okay, homozygous, 2 identical alleles, heterozygous, 2 different alleles. When we look at genotypes, we have different ways to say those: homozygous dominant, homozygous recessive, heterozygous. These are just slides that you can test yourself with as you're looking. Okay. Now, everything we've talked about so far has only been true for traits that display something called complete dominance. So we've only talked about complete dominance so far. That's where these traits have one dominant allele that masks the other recessive allele. This works because the dominant allele actually codes, so remember your DNA codes for proteins. Dominant alleles code for proteins that work. However, recessive alleles do not work. So if you think about flower color, purple was dominant. That's because the purple flower color was being made. If you have a copy of the genes that produces a working protein or a working purple color, then you're going to have it. Whereas the recessive allele is actually a broken code, so it doesn't make the protein. So we had white flowers. So that's just kind of a neat thing to remember. But not all traits are controlled by complete dominance. There's some other things such as codominance. Codominance means there's no dominant or recessive allele. Both alleles are expressed fully and at the same time. They both code for a working protein, so there's no broken recessive allele. They're just two different versions. An example of that would be blood type. You have A blood, you have B blood, you have AB blood, and you have O blood. So in A, we have one type of protein being made. In B, we have another type of protein being made. And then in AB, we have both types being made. Neither is recessive over the other, just both occur. O is actually where no protein is being made. And this has to do with receptors on the blood cells. Okay, and then there's incomplete dominance. In incomplete dominance, one allele is partly dominant over the other, leading to a blended phenotypes. If you guys have ever seen snapdragons, there's these really cool flowers. There'll be red flowers, there'll be white flowers, and then the hybrid is not red like you would expect. It's actually pink. It's a blend. So less red is made in the incomplete dominance. So there's just less of the red protein being made, which makes it appear pink. Okay, that's incomplete dominance. I'm going kind of quickly because I don't have much time on these. We also have to look at gene pair interactions. This can lead to unexpected phenotypes like these Labrador retrievers. One really cool thing is black labs are actually dominant over brown labs, but then what happens with this yellow lab? Where do yellow labs come from? So we have dominant, we have recessive. Well, what's going on here? It's actually a totally different gene called the melanin deposition gene. So melanin is going to be the pigment, the coloring, and this gene E here controls whether melanin of the coloring ever even gets into the hair follicles. So we have, for example, this is a dihybrid cross. Dominant E means that the hair, the coloring is deposited into the hair. So we have right here, there is going to be coloring because we have dominant alleles, and that coloring is going to be black because that's dominant. If you look at this one right here, there is going to be coloring because we have dominant alleles for E, but it's going to be brown because that's recessive. Now look down here. These are recessive for E, homozygous recessive for E, meaning that no matter what color this other allele is, it doesn't matter because none of that color is ever even going to get into the fur, making it a yellow lab. So that's pretty cool. And then there's something called pleiotropy. This is when a single gene affects multiple traits. One gene, multiple traits. For example, 40% of cats with white hair and blue eyes are also deaf. You wouldn't think that they would have anything to do with each other. However, they do. Somehow those genes are intertwined. They actually affect each other. And the same thing with sickle cell anemia. It causes, we're going to talk a lot more about this later, but sickle cell anemia causes a sickle-shaped red blood cell when oxygen levels are low, which can cause very serious health problems in homozygous recessive individuals. However, heterozygous and homozygous recessive individuals are resistant to malaria, so that's pretty amazing and beneficial. And we're going to talk more about sickle cell anemia later on in another chapter. So make sure you go through both Mendelian complete dominance, as well as codominance, incomplete dominance, gene pair interactions, pleiotropy, and know the difference between all of these when you're looking over your slides. I will make some more of these for the next chapter, and I miss you guys in class. Let me know if you have any questions.